So at this year ASH meeting, we had what I really think is really the next chapter on the immunotherapy revolution in multiple myeloma. The last few years, we have been bombarded with very exciting data about BCMA targeting approaches, either antibody-drug conjugate, CAR T cells, and bispecific T cell engagers. At this meeting, we saw more of that. We saw several different CAR T platforms. We saw bigger and more mature data from the CARTITUDE study. And we saw more mature data from the KarMMa-1 study. But to me, what was the most exciting is we're learning about a new array of bispecific T-cell engagers that use targets other than BCMA. So we learned at the meeting about Talquetamab, which is a dual body bispecific T-cell engager against GPRC5D. And that's the first time that we have any clinical data that shows efficacy of targeting that particular molecule on the surface of plasma cells. This was a phase I study, and at the highest dose tested, patients are achieving more than 70% overall response rate with a drug that is quite well tolerated with essentially only grade 1 and 2 CRS on the first few doses and can be administered subcutaneously. So that's extremely exciting. We also invalidates a novel target that I'm sure is going to be pursued with other platforms, including CAR T cell. We also learn about Cevostamab, which is also a T cell engager, bispecific T cell engager that targets FcRH5. This is administered for the time being intravenously, but the signature convenience feature is administered every 3 weeks, as opposed to every week as most bispecifics. Again, at the highest dose tested, we saw about 2/3 of the patients having a response, and most of the toxicity being, grade 1, grade 2 CRS. So those two really open two new frontiers, I believe, in myeloma that will be pursued, aggressively in terms of sequencing combinations. It opens all sorts of questions and opportunities for myeloma therapy. And we saw very more mature data with Teclistamab, bispecific BCMA, that is also a dual body that shown now with more mature data with both the intravenously and the subcutaneous formulation. Again, very convenient for patients, also showing a very high rate of response and excellent safety profile. And on the same end, with several others, including AMG 701, which was the former AMG 420, but now with a half-life extender that allows it to go from intravenously to, from continuous infusion to once a week administration, also showing excellent activity in the higher doses and several more. So I think this is definitely the ASH of the bispecific T-cell engagers.